Does it work? Yeah, good morning. Yeah. So, happy Saturday. This is, I always worry a little bit when I give a talk on a Saturday morning, especially if we had drinks the night before. But it's that's my story with the ULF. Anyway, I'm Ali Fatemi. As mentioned, I'm a pediatric neurologist at Kennedy Krieger, and have been working on the leukodystrophy since 2001. So most of my adult life, since I'm only 25. But this is intended to be for lay audience, and it's really to focus very roughly on the different strategies that we're currently employing to try to treat these difficult diseases. And I'm not going to talk about any particular drug, well, maybe a little bit, but um, I do have a little bit of a disclaimer that I'm myself an investigator involved in multiple clinical trials in different diseases, and also have developed some drugs myself that are patented. So just so people are aware. But I do think that there is a huge amount of hope. So back in 2001, when I first came to the ULF, we had a handful of therapies that we were considering, but really the only thing that had reached clinical trial stage and for which we had data was something we call bone marrow transplantation, and that was for adrenoleukodystrophy. And that has become standard of care now for many years. And I'm not going to talk about that because that's standard of care. So I'm going to talk about new things that have emerged over the last couple of decades. But when we want to talk about treating a disease, and this is a challenge that we have, our biggest challenge is that we have to actually think about what are we trying to achieve here. And so when we talk about a clinical trial, it would be good to just think about what do we actually mean with a clinical trial. So a clinical trial are trials that involve research using people and advancing clinical knowledge. And there's different types of studies that you can do. There are some studies where you don't even give them anything. You're just observing what's happening because that's critical. If you want to treat a disease, you need to understand first what happens if you don't treat the disease. And the rarer a disease is, the harder it is to know what's happening when you don't treat a disease because there's fewer people that can give us that information. And then you can try to first go into a human to what we call a phase 1 study. So these are the very early stages when you try to see what happens when you give something to a patient. Is it safe? Do they get a big allergic reaction? Do their faces swell up? Do they start vomiting? Can they secrete the drug? How long does it stay in their body? What concentration does it achieve in the blood when you give them this dose? So these are what we call dose finding studies and safety studies. Those are the very first steps that we do. And that can be a handful of people and sometimes this is done. And until very recently, the regulatory agencies required that we actually do this in healthy people. It no longer really makes sense with the strategies I'm going to talk about because some of those things you just can't give to healthy people. They're very selective for the patients with their condition. And then as we move on to really try to understand whether these drugs are working or not, we, for statistical reasons, just purely due to math and the probability of chance, we need more and more patients. And so in a condition like, say, hypertension, high blood pressure, right? I have high blood pressure, the drugs that I take, those trials to get approved, they tested 20, 30,000 patients. And they do that because there's a lot of people with hypertension. 30% of the US population has that. And at the same time, there's so many things that can affect it. How much coffee you had before, how much alcohol you had before, et cetera. So you need to have big sample sizes to take care of variability. And so that's a big problem in the leukodystrophies because these are rare and ultra rare diseases. And so these are all the big issues that we start off. And then sometimes what happens is you get a drug approved and they may have some long-term side effects that we don't know of. And they won't show necessarily until decades later sometimes. And so even once a drug is approved, there are oftentimes the so-called long-term post-marketing safety studies where we try to see in the long term if there is an effect that we didn't wish to have. And so when you want to do a trial, you have to really think, what are you trying to achieve? We all want to be better, right? But what does that actually mean? Does it mean that we want to just stop a process that's getting worse from getting worse? Or do we want to actually reverse that process and feel better? That's the very first question. What's the goal? Um, and then there is a big problem, and it's a good problem that we have, but it's a bad problem when it comes to a clinical trial, and that is the power of our brain. Our brains are really important in the healing process, and that's called the placebo effect. This is a nature's way to protect us from dying from every illness we have. We have an amazing regenerative capacity that depends on our mind. And that's as strong as 30 to 40%. That means that when you take something and you think it's going to help you, there is that level of a chance that you're actually going to feel better. And so that becomes a big issue when you try to actually see if your drug is working. And you also have to think critically about what kind of patients you're going to try this on, because if people look too different and you try the same approach to them, you're not going to be able to compare those data points with each other. And so I want to talk about the placebo effect because that's been the biggest challenge in the rare disease field. And so this is an example of what I mean with placebo effect. These capsules are fabulous. When I look at the box, I stop coughing. Okay, so that is what it is. It is the fact that we feel better because we want to feel better. And it messes up your intervention. And so in order to avoid that, the ideal clinical trial design is what we call a placebo-controlled double-blinded study. A double-blinded study, hypothetically, would mean that I don't know what I'm giving you, and you don't know what you're getting. And there is a sugar pill, and there is the real pill, and they look identical. And then you'll take it, and then we'll figure out if you're getting better or not. And after a treatment period, then we sort of unblind it and try to look at and see if that effect is actually real or if it's just a perceived effect. And there are many ways that these things can become complicated. For example, when we do a treatment trial where the outcome is survival. So when we're doing gene therapy, say we want to see if the patients live longer, right? One of the things that we know right now is with modern ICU type of care, we can keep people alive for a long, long time, right? Because you can be on a ventilator, you can be on a feeding tube. You may not do anything, but you can be alive for a long time. And one of the big questions is that a decision that you're doing because you think, oh, well, my child is on this drug, and because he's on this drug, maybe we should keep going? And then it becomes sort of a perceived notion that it's the drug effect, that the survival is longer. But in fact, it could be just a parental effect that they thought that it was working. So these are challenges, that are ethical challenges that are there that we have to manage. And us doctors are the worst. We are the most biased. When we hear about a new therapy, we get all excited. We want to think that it's working. And so it's really also important to try to keep us blinded as much as we can. But it's all easier said than done, because in the reality, when we get to trying to do a clinical trial, and we want to do what we call the gold standard, what I just mentioned, is double-blinded placebo-controlled trial. But that's exciting and that's great, but it's not practical, and in some cases it's not possible at all, right? When you have 5 patients in the world, it's not going to be possible to do a placebo-controlled trial. If you have an intervention where you are giving a person chemotherapy, wiping out their immune system, and then giving them the drug, well, you can't give somebody chemotherapy and wipe out their immune system and then not give them a drug, right? I mean, that's unethical. And so then you can't really randomize the study. So you're basically having to select. And so what comes then at play is that it's really critical to try to understand what is the course of a disease without any treatment. So that's what we call a natural history study. That's why doctors keep bugging patients and asking them, how did you look like when you were 3? And the guy's like 83, right? So because we really need to understand how does the disease course change. And that's the thing that we still know the least about, because we have all these new diseases that we have discovered in the last 15 years. And we don't really know how variable they are. You can go downstairs and look at the kids here, and they may have the same disease and may look very different, very different. Sometimes even siblings may look very different. And so these are challenges that we have to overcome. And so we try to do placebo control as much as we can, and we try to what we call randomize people. Some of the tricks that we try to use in this field is, for example, to randomize in a way that instead of giving one pill to one person, one pill to another, one sugar pill to a person and one actual pill to the other person, we randomize them in a skewed fashion. Meaning, for example, thought it was a third. Oh yeah, here. We try. This would be ideal for rare disease. One person gets the sugar pill, 3 get the actual intervention. So that way, when you get enrolled in a trial, you have a much higher chance to actually get the intervention. Another trial design that sometimes is used, if it's making sense, is what we call a crossover study, where you have 2 groups of patients. One goes on placebo, the other one goes on the drug, and then after a period of treatment, you switch them around, and so that's only possible if you can measure something that can change within that period of intervention. Okay, so I talked a lot about design, but let's get into the actual stuff, right? So what are the ways we try to help people? We're talking about genetic diseases here. These are leukodystrophies. So that means it all starts with an abnormality in a gene, right? So we all have, I don't like standing there. Okay. We all have 3 billion DNA letters in each single cell in our body. And when we have a genetic disease, that means that there is something wrong in one of those 3 billion DNA letters that results in a disease condition. And so that abnormal genetic finding ends up making a protein that is abnormally functioning, meaning it's either not functioning or doing something it's not supposed to do, or in some cases, when you have a mutation that results in not having this protein at all. And that then results into a leukodystrophy, okay? So when we try to fix this, there's different ways we can approach it. One way is to try to go and fix this genetic defect at the genetic level, so at the DNA level, at the gene level. One way is to try to do something to the protein to make it function better, or replace this missing protein and give the patient the actual protein. One way is to say, okay, this protein is there, but it's not functioning well. Let's give it something to work a little better. Or sometimes when all of this has failed and the patient now has the disease, we try to come with what we call a restorative approach, meaning, so, oh, well, these cells died. Let's try to replace them with new cells. So that's sort of the hardest part. So there are strategies where you try to sort of prevent, and there are strategies where you're trying to restore, and they're a little different, and restoration is always much harder. So most of the treatment approaches we have out there are currently trying to stop the disease from getting worse rather than trying to actually reverse symptoms. So I don't know why I had built in this thing. So let's talk about actually trying to do something at the genetic level. There are different approaches that are in clinical trial stage and new technologies that are emerging, and we're going to talk a little bit about those. So when you have a change in your gene, we used to call them mutations. Now we speak more of pathogenic variant because there's this perceived notion the word mutation is not a good, whatever, not a politician. So that can result in multiple things. One thing is that change in the gene can actually be silent, meaning it doesn't do anything. So any person in this room, we take our genome, we look at it, we all have different DNA letters, and we have these variants, and each of us has hundreds, if not thousands of variants within these 3 billion DNA letters. And most of them don't make us really different. I mean, they make us different all individually, but they don't make us sick. But you can have a change in the gene that results in no product at all, meaning that DNA is so abnormal that you don't even get that protein, the product of that gene. Or you may get a protein that's less active. Or you may get a protein that is not doing what it's supposed to do. Maybe it's supposed to do something like breaking down a product, but instead of breaking down that product, it does something else. And so that's what we call a gain of function or a toxic mutation. So this is a change that results in a product that does something that is not good rather than not working. So it's like the bad soldier, I guess. Or you can have too much of a protein, and if you have too much of something, it's also not good. So biology is very, very fine-tuned. Life is a very, very fine-tuned situation. It's called a fine-tuning problem in physics. So if anything is too much or too little, that can result in a situation. And so the different approaches that you can change that. So one is, if you don't have a product, or if you have a product that's not working well, you can try to just replace that. So for example, adrenoleukodystrophy, there's about 1,500 changes in the gene that we have discovered so far. And all of them are what we call loss of function mutations. So either they make no protein, or if they make a protein, that protein is just not working good enough. And so one of the strategies that has been chosen is to basically replace that by giving the patients a product that has the actual correct gene in it, and then let the body do its job. So that's what we typically, when we call something gene therapy these days, that's what we're talking about. But then there are also other things. It's like, what if you have a disease where you have a product that's misbehaving? So there's a disease called Alexander disease, and there are changes in a gene called GFAP. And these changes are felt to be mostly what we call a gain of function mutation, where that GFAP is abnormally functioning. And so we want to get rid of that GFAP. And so that's a strategy where you either try to silence that gene, meaning shut it down, or you try to actually go in and correct that sequence. That's called gene editing. That's like, taking an eraser, wiping out that abnormality and putting in the right sequence. So we're going to talk about all these right now. So let's talk about regular gene therapy or gene delivery, right? So this is just giving the patient what they're missing. And so you can do this two ways. We talk about in vivo or ex vivo. In vivo means you're injecting that missing gene directly into the human body. And depending on where the disease is, you may have to inject the gene in different places. For example, you may give that gene just through putting that gene into a virus and then injecting that virus into the bloodstream, right? And then try to see if that virus makes its way to the brain or the spinal cord or wherever it needs to go. Why do we use viruses? Because that's what viruses do. Viruses, basically, their job is to deliver genes into the body. And we take viruses that are usually infectious and are usually dangerous, and we modify them in the lab. We make them less criminal, we make them less dangerous, but they are still infectious, so they can still infect your cells, and that's what we actually want to do. But instead of bringing in their own DNA, they're now bringing in the DNA that you want them to bring into your cells. Ex vivo is a similar strategy, but here you're not actually injecting the virus into the body. What you're doing here is you removing cells from a patient into the dish, for example, bone marrow stem cells, hematopoietic stem cells, and take it from the bone marrow into the dish. You fix that genetic abnormality there by giving them so you're not editing here. You're basically just infusing them back cells where you have treated those cells with the virus that has the gene inside. So for example, there is gene therapy right now approved for adrenoleukodystrophy and metachromatic leukodystrophy, right? These two gene products are basically viruses called lentiviruses. And what they do is they take the patient's own bone marrow stem cell, they treat those stem cells with these viruses in the dish, and then they give them back their cells. In vivo gene therapies, again, is different. There, you're directly injecting that virus, and it's either in the brain or into the spinal fluid or into the blood. And let's talk about that a little bit. So delivery is actually the biggest challenge we have right now. It has become pretty easy to develop a virus that has your gene packaged in it. The challenge is to get that virus to the place where you want it to go. And there's two challenges. One is that A, you got to get your virus to where you want it to go. B, you have to make sure that it doesn't go to the places that you don't want it to go, right? Because if we get a gene that is supposed to be only turned on in the brain, and now we give that gene to the whole body and it goes to your kidneys, it may do the wrong thing in the kidneys. Again, the body is very fine-tuned, and it's fine-tuning problems. So we can't just be like, oh, well, let's just give it somehow. So many times we have to really think about that, and different routes may have different patterns of side effects. And so a big problem is the brain, because our brain is huge. It's the universe's most complex organ, as far as we know. And getting a virus to spread through the entire brain is not trivial, and is one of our biggest challenges. And another problem is that we talk about viruses. And so guess what? When you get an infection, why do you get a stuffy nose? It's not the virus itself, it's your body fighting the virus, right? That's the immune response. And so what our bodies do is when they get the exposure to the virus, they start building antibodies against it. Now, it may not matter depending on how you deliver it. If you deliver it into the brain directly, for example, you may be privileged in a privileged site, meaning that the immune system doesn't get into the brain as much to destroy that virus. But there are also viruses that are where they're common viruses, and so we may have already antibodies against those viruses by the time we get them. So these are all challenges that we face. But again, the biggest one is the size. So we do things in the lab with a mouse brain, and that's a mouse brain. It's actually bigger than a real mouse brain. The real mouse brain is about the size of my pinky, like the tip of my pinky, right? So then you see the rat, the ferret, the monkey, and you see the human brain, how gigantic it is compared to that, right? And so you may, here you have tiny amounts of white matter, and you may be able to inject one shot into the brain, your virus, and it spreads around easily because it only has to walk like, 0.2 inches. You inject one little thing here, it's only going to stay local. It's going to be really hard for it to spread. So there are challenges in terms of delivery that require a lot of engineering, meaning engineering of that virus to make it more potent to be able to spread further. And there's some tricks that we can do depending on where in the brain we inject things to take advantage of some of the pathways that exist in the brain for the viruses to sort of travel along. Another technique that it has become important is the other way. For those mutations where we're trying to silence the gene, where we have too much of something that we want to get rid of, right? And so that's called silencing. And so there is a therapy called ASO. And so what does that actually mean? So go back to very basic genetics. So again, we have our genes are inside the DNA, right? And then in order for this information to be turned into a product, the protein, these genes are first transcribed into something called the messenger RNA. And the messenger RNA is basically a photocopy of the DNA. It's a photocopy that is taken from inside the center of the cell and then taken out. And then that photocopy information is read by the cell to provide information to make the protein, okay? And so this gives us an opportunity because we could try to go in and use a photocopy of the photocopy, okay, to try to block this thing. And so as a consequence, we block the messenger RNA. Now, depending on where you block the messenger RNA, and that's critical, depending on where you block that messenger RNA, you may either suppress the protein synthesis, or in fact, there are some areas called regulatory sites that are usually helping to silence too much of a product, and you can block the silencers. So if you block the silencers, they become loud, right? Meaning you make more protein. So you can use this strategy to increase or decrease protein synthesis, depending on your gene and depending on your gene mutation. And more importantly, sometimes this can be very specific to a single patient's mutation. Okay, so this can become what we call an N of 1 approach, where you take an antisense oligonucleotide that's targeted very specifically against that one patient's variant in the gene. And so there are some advantages of this. This is cheaper to make than those viruses. The sort of one advantage or disadvantage, depending on how you see it, is gene therapy is considered a one-time shot. When you get that virus into your body, you can't get that virus again, because remember, you would build an immune response potentially. So you do it one time, and the idea is that that gene is going to stay in your body for the rest of your life. Here, this drug, this photocopy drug, lasts in your body for a few weeks to a few months. It's eventually cleaned up. It's eventually secreted or cleared up by your body. And so the advantage is that if you have bad side effects, it will be gone eventually. The disadvantage is that you have to keep doing it every few weeks to every few months. The biggest problem with this approach is that this stuff, the cells love to chew it up. Okay, so you really don't have a lot of opportunity to keep that thing. If you inject ASOs into the bloodstream many times, they never make it to where you want it to make. They're cleaned up by the body before they even get to your target organ. And so delivering these things, again, is a challenge that requires usually either injecting into the brain or into the spinal fluid. But this is again an approach that's been well into clinical trials. We have approved product. We have in spinal muscular atrophy now, ASO therapy that's become pretty much standard of care. And there are clinical trials right now in Alexander disease and other diseases, leukodystrophies that are happening using this approach. And in Alexander disease, remember I mentioned earlier, there is a mutation in the gene called GFAP that makes this GFAP misbehave. So you're trying to silence it using this strategy. Then I want to talk about the more recent stuff. This is just getting into the clinical trial stage. This is not in the leukodystrophies yet, really happening at the clinical trial level. I'm hoping within the next few years, we're going to have our first trials. And that's CRISPR. I mean, there's been a lot of talk about this. I know you've heard a lot about it in the news. There's a lot of companies that have emerged with billions of dollars of investors over the last 10 years about this. There's a little bit of a hype too. But what this is really about is to try to not just give the patient an antisense oligonucleotide or give them the gene that they're missing, but actually going into your DNA and fixing that very abnormal defect. So you're going in there with an eraser, you erase the mutated part and you replace it with a normal nucleotide. And so that's why it's called gene editing. It's editing the actual gene. And I'm not going to get into this, it's maybe a little too complicated, but it's basically a very ancient system that we take advantage of from an evolutionary perspective to go in directly, find the area that is abnormal. We use something called the RNA guide that can find exactly where your mutation is. You have to know what the mutation is. And then there is an enzyme that goes in and chops it out, and then it's replaced by the body with the correct copy. And so this is awesome, because now you can actually go and fix the genetic defect for good, actually correct it at the DNA level. The disadvantage is that this stuff is big. So getting this enzyme and the RNA guide into the body requires a carrier that is currently a little too big to fit into the standard viruses that we use. We're working on it. It's a lot of stuff that's been happening in the last 5 years to make these what we call base editors smaller and smaller so that they can fit into the viruses to get into the body. But it's still the main challenge. And so that's going to change, I think, in the next few years. But we are about to hit clinical trial stage for the leukodystrophies here soon. And this has been used in some other diseases, for example, in sickle cell disease, where you are taking, again, the patient's bone marrow stem cells out of the body, and then you fix them with the gene editing in the dish, and then giving those back to the patient. So you're not injecting this stuff into the patient, you're injecting into their cells outside the body and giving it back. And then I want to talk about more simpler therapies, let's say. I don't know if the word simple is appropriate, but small molecules basically are not going at the gene level necessarily, but what they're doing is these are usually pills that you can actually take. Sometimes they're infusions. And what they're doing is try to correct something that's abnormal in your metabolism or in your pathways in your body. One example is, for example, there is a small molecule used for CTX. So this is a disease where you have a defect in this cholesterol pathway, and as a consequence, you end up having low cholic acid and chenodeoxycholic acid levels. And so you can basically give the patient the chenodeoxycholic acid as a supplementation. And there's some studies that a lot of this data is coming from Italy that shows that it does have a pretty significant impact on the patients. It's just to give you an idea, this is just a small, chemical, right? And so as opposed to what I was talking about, the DNA and RNA and so on. Another small molecule that's right now in clinical trial stage is called ISRIP. So this is the vanishing white matter disease is a disease where you have mutations in one of 5 different genes, and these 5 genes build a complex called the EIF2B complex. And so this complex has a core in the center. That's what we call the active site of this enzyme. That's and it's regulating the so-called integrated stress response. We don't need to get into that detail, But what we do know is that there is a little drug that was developed. It's a small molecule again that can go and stabilize this complex. And so this is currently in clinical trial for vanishing white matter. And again, this is a pill that you can take, and it's done as a small molecule therapy. And then I want to talk finally, and I'm almost coming to an end, about another approach that's sort of, and we talked initially about restoration, right? So sort of the Hail Mary kind of approach is like, well, you have now somebody who already has a lot of dead cells in their body. They lost all their myelin, their neurons are dying off. Is there anything we can do still to restore function? You know, this is like the sci-fi kind of approach. And it was really sci-fi until around 10, 15 years ago. And then a big discovery that was made is that you can take any cell from your body pretty much and reprogram them back to become like a stem cell. So you can take a skin cell, you can take a blood cell, and you can treat them with a bunch of things that basically different factors that reprogram these cells and make them behave like a stem cell. And so these are called induced pluripotent stem cells, iPSCs. And then you can take these iPSCs and you can differentiate them into pretty much any tissue type you want, including myelin-producing oligodendrocytes or neurons even. And this has become actually a very big thing in research. Like a lot of our research these days, rather than using animal models, is actually using human patient-derived iPSC cells that we turn into neurons. In my lab, we have these, what we call cerebral organoids. Those are basically, I don't know if I would call them mini brains, but people call them mini brains. These are basically these little, point size organoids that are derived from these iPSCs that include essentially all the structures of the brain from our patients that we took from their blood. So we took the patient's blood, turned it into a stem cell, and then turned them into these sort of mini brains. And so those are great models to study what's going on in these diseases and test therapies. But the other thing you could theoretically do, is turn these stem cells. So say you have, for example, adrenal leukodystrophy. You could take a patient's blood cell, you could turn it into an induced pluripotent stem cell. Here, you could fix the genetic defect in a dish. And then you can turn those cells into their oligodendrocytes and neurons. And then you could try to inject these cells, these stem cells or progenitor cells, back into the patient's brain. Again, it's a little bit of a sci-fi because it would require these cells to know what they're doing. When you inject them in a space, they need to know, and they need to now to go and grow and integrate into the network. And so the reality is that that's really not happening yet. There have been clinical trials using similar strategies already in the leukodystrophies. For example, in Pelizaeus-Merzbacher disease, more than 10 years ago now, there was a trial where neural stem cells were injected directly into patients' brains in 4 patients. It was 4 or 5 patients, the study that was done, to see if they can turn into myelinating oligodendrocytes, because that's what's sort of wrong in that disease. And one of the patients died, actually. And in the autopsy, they could see that these stem cells had differentiated and had started making myelin, but they hadn't spread far enough. And again, part of the challenge here is this issue that I showed earlier, big size. Like if I inject cells here, they have to make it all the way, and it's like basically trying to walk across the world, across the globe. If you're a stem cell, your human brain is the size of of the Earth, and so it makes it really challenging. But it's coming, and people are keep working on these strategies. People are trying to say, well, maybe that's too far, but maybe we can make, for example, cells that are not necessarily integrating themselves into the brain's network, but maybe they can release factors that support the brain to regenerate. And so those strategies are being actively discussed. And then I just want to end here by, is this? I don't think it's the last slide. Oh, I thought there was one last slide. Yeah. So, if you're a patient and you want to know if for your disease there is a clinical trial, you go to clinicaltrials.gov at least in the United States and most European countries, it's required that every clinical trial is registered with clinicaltrials.gov. So you have, if there is a trial happening in your disease, it will be on that website. It can be a little confusing because again, let's say adrenal leukodystrophy, you type that in, there will be 50 different things that are listed. Some of them are saying not yet recruiting, some of them saying no longer recruiting or completed enrollment. If you're interested in a clinical trial, you want to look at the ones that are saying recruiting or the ones that are saying not yet recruiting, which means it's probably going to start recruiting within the next few months. So that's the biggest sort of source of information if you're interested in clinical trials. And then usually they have a contact information for the study director. They list usually what we call inclusion exclusion criteria are. That's the place to go. So I'm going to stop here and use the rest of the hour for questions. I know this was a lot to cover in a short amount of time. Go ahead. Replenish white matter. Uh-huh.
And you say to inject that into one place, it's got so far to travel, but has it been considered to put it in multiple locations?
Yeah, so the question for those online is, if you want to replenish missing white matter, yes, injecting in one place is not enough. Can you do it in multiple places? And that's exactly what actually these trials did. So that trial that I mentioned on Pelizaeus-Merzbacher disease, they injected, I want to say, on 2 or 3 different injection sites on each side of the brain. So that's a lot of injections, right? But unfortunately, that was the strategy that has so far been pursued. In the last 5 years, there is a group that's been, at least one, I'm sure there's more, but I know of one group that's been using a different approach where they have taken these stem cells and have modified them further, so engineered them further to allow them to be able to better cross from the blood vessel into the brain. And now they're working on intra-arterial delivery. So you have 2 big arteries here in the front, right? The carotid arteries, they supply most of the blood to the forebrain. And so the idea is, rather than trying to do brain surgery, you do vascular surgery, you basically inject into these arteries, inject these cells. The problem is, like, again, they may not get into the brain because they have to cross from the blood to the brain. But you can engineer them and give them a little receptor that binds those transporting mechanisms to try to get them into the brain. So that's something that's being worked on. And if that works, that would be neat, because then you could do an injection here and get a global delivery to the brain rather than multiple injections. But now you're bringing in additional layer of complexity with potential more side effects, because you're taking a patient's cell, turning it into a stem cell. You already genetically modified it to do that. Then you're fixing their genetic defect. Then you're genetically further modifying them, giving them something that they shouldn't have to allow them to do this migration. And so each of these steps increase your risk that these cells might misbehave, right? And so the biggest risk we always have with cell therapy is that they may do something you don't want them to do, or they may turn into a tumor cell. And so that's actually a big concern. The good news is that we haven't seen these cells turning into tumor cells really yet since the iPSCs come in patients. We've seen this in animals. So there is that concern in the animal side. But in the old days, when the very first stem cell therapies were started, this is like early 2000s, so the old days, that time, before the iPSCs, there were cases of brain tumors that resulted as a consequence of injecting stem cells. Now, the reported cases were usually in, how do I say that? Maybe shady places where these trials were performed. We didn't really know what these stem cells were. They weren't necessarily under a good regulatory agency. So there's a lot of things that we can do to monitor these, but that's always a concern with stem cell therapy, is that those cells could turn into some sort of tumor cell or could attack the body, that kind of stuff. But the idea is there and is being pursued. So I think, I'm looking forward to it still. Now, I gotta just admit, in my own lab, we worked on stem cell therapy for like 7 years. And I moved away from it because it was getting really hard to translate it into a clinical trial due to these challenges, and I feel like, you know, gene therapy is actually easier. These viruses are easier to spread, much easier than the cells. The cells are, you know, thousands of times bigger than a virus. So in comparison. Yeah.
With CRISPR technology, you ex vivo create genes that are appropriate, that are working well, and then you put them back into the body. But how is it that those few genes can replace all of the problematic genes in the body? You know, in a condition like CTX, every single cell in the body has the faulty gene.
Right.
How does that replacement happen?
So it doesn't, and that's the problem. So right now, the CRISPR technology is mostly applied in an ex vivo setting, where what is happening is what you said, is like you take a set of cells, like bone marrow stem cells, you fix the gene in the dish, and then you give them back. And obviously that's not sufficient, because you need to get that to every cell in the body, and it doesn't get into every cell of the body. And so the biggest challenge right now is what we call in vivo gene editing, meaning trying to edit these cells in the brain, in the body. And in fact, the NIH has a, you know, they call it a $6 million you know, price if you can do that. So there's a lot of effort, like I said, trying to make that happen by trying to shrink these base editors to make these proteins smaller and smaller, so that their genes become smaller and smaller. And then you can take their genes, stick them into a virus, and then infect the patient with the virus, and to spread that as far as you can. The other issue is that one of the things that we talk about is usually editing efficiency. So these enzymes that are called the editors, so the ones that actually go and edit and change and fix the genetic defect, they don't have 100% efficacy. So at best, when you look at studies in vitro, so even in a dish, when you have direct exposure, we see sometimes, you know, if it's awesome, it's like 95% efficiency. So you never get to that 100%. But it's also important to say that it really depends on your disease. For example, take a disease like metachromatic leukodystrophy or Krabbe disease. Right now, some of the thought processes there are that even if you don't correct the MLD gene, it's called, you know, our sulfatase, in every single cell, if you correct it in certain cells, then that enzyme is produced in those cells, and the enzyme is then transferred from one cell to another cell. So you might still be able to spread that correct gene across the body, just simply because cells communicate with each other and transfer certain things. But not every gene can be transferred from one cell, or not every protein can be transferred from one cell to the other. And so, for example, in adrenoleukodystrophy, there's no evidence that adrenoleukodystrophy protein can be transferred from one cell to the other. It's more common with lysosomal storage disorders like Krabbe disease and MLD and so on. So it really depends on the gene and your disease. Yeah, hope that helped. Did you have, the question? Yeah, go ahead.
Thank you for a really informative session. So my question is regarding the strategy for monogenic gain of function gene therapy, monogenic gain of function. And sometimes, like Alexander disease, you want to lower the levels of GFAP, but you don't really have a fine control over how much you can knock down. And sometimes all those things are required for, let's say, cell integrity and other functions required for life-sustaining things. How do you approach that challenge?
That's a big challenge. And as my biggest concern with ASO therapy for those diseases is that there is really nothing in nature that's useless. Okay, so every single gene has probably many functions. We know usually of one, we call that the canonical function, but we're learning now that each gene may have numerous functions at different points in life in different cell types and so on. So suppressing a gene is probably not a great idea. And there isn't really any way we can sort of calculate and say, well, if we give him this much, then it's going to be this much of a suppression. That the only way to figure that out is to do animal studies and get a guess. And then really the only way to figure it out is doing it in patients. And so the problem with that is you need to have a good biomarker that tells you what's actually going on in that patient, right? And so that's what we are really missing in the many diseases.
Right. And the translation between the animal model and human
Exactly,
is very
exactly.
limited.
Right, right.
And then for these potential detrimental effects to show up, it's not just immediate after the injection. It's going to take a long time for it to
Yeah,
actually
yeah.
show.
Yes,
So
and then the other issue is that these are often, again, specific to a patient mutation. So you can't even study these things in like healthy people, and you may not also be able to study these things in animals because the sequence of the gene is not the same in an animal. And so these sort of toxicity studies with ASOs are always challenging because of that. And that you really don't know what to expect until you inject it into an actual patient. And you might get false toxicity. That's part of the problems. Like, you know, you might inject it into healthy people, and there it might cause a problem because there you're now bringing it down to 0. But in a patient who has too much, it wouldn't go down to 0, right? And so really you can't use a control even to test this. So you have to go to a patient. And so many times the regulatory agencies then say, well, if that's the problem, then you can only do this in a small number of patients who would die otherwise anyway. And so let's at least, you know, try it in that cohort. That's oftentimes what ends up happening. And then in those patients, it's often difficult to really know if it's helpful or not, because they're usually severely affected patients, right? But it does give us an idea about, you know, a little bit of an idea of what we call pharmacokinetics and dynamics in terms of what they're doing. But I would say the truth is that, for example, in Alexander and in PMD, these things are not potent enough, really, as far as we know, to really bring it down to 0. So it's very unlikely that this is going to really go down to 0. And we also know that nature has some resistance. And so many times you might be fine even if you have like 5 or 10 or 20% leftover function. So you may not need like exactly, you know, 100% of your protein.
we just don't have the control. But
Exactly, exactly.
thank you. And astrocytes are dividing cells. So my next question would have been, how would you anticipate the persistence of efficacy after injection? Sorry, this is about gene therapy, obviously.
Yeah. So you're talking about gene therapy or ASO?
Actually, could be both, but astrocytes are dividing cells. So mostly, I guess, gene therapy.
Mm-hmm.
And we don't have enough data to actually calculate how long the efficacy
Right,
will last.
right.
And then we have the situation where we cannot top off at the moment.
Yeah.
So it's a 2 question. One, do you have any advancement in the technology that you can top off, for example, changing the capsid type or changing completely the viral vector type or something like this, or silencing the immune system altogether, like in chemotherapy, to make the second dose possible?
Yeah. So people have not gone in nervous system diseases. People have not gone the route of second dosing yet. That discussion is emerging in some of the liver diseases, the hemophilias, where you have a blood clot factor that's missing, and you have to inject into the liver. So many patients are more likely to develop antibodies against it, and that's actually, you know, I don't know what I can say, but that's being actively pursued to figure out a way to slightly change the viral, you know, vector to avoid the immune response and to try to give the gene, you know, every few years. So that in the hemophilia world, that's actually probably going to be needed. Is that every few years they're going to have to just get another round of gene therapy with a slightly different vector that avoids the you know, antibody response. In the CNS, this has not been pursued, but also a lot of the gene therapies in the CNS, again, are directly targeting the brain, and so they're less likely to be affected by the immune response.
I agree, but the conditions like Alexander disease, the BBB is affected because astrocytes are
Yes.
part of the BBB.
Yeah.
In that case, there's
Yes, there's more exchange. Yep. Yeah, totally. Yeah, that's a problem. And you know, the one of the factors that's always hard to know is in a replicating cell, what percentage of the gene product will actually remain in the you know, down the road. So you know, there's sort of 2 ways to think about it, is that on one way, you would be like, oh, so maybe we should use viruses that integrate into the genome, so then they're built in there. The downside of that is if they integrate themselves into the wrong site, then that can cause cancer. And we're seeing that, for example, now in adrenal leukodystrophy with the lentiviral. And so nothing is perfect. No, sorry, but that's where we are. I know maybe somebody else has a better answer in the room than I have, but I think we are still struggling with all of these things. And that shows you the reality of the complexity of doing these sort of advanced genetic therapies.
Thank you.
With personalized medicine coming in, like how do you do a clinical trial if n equals 1? I'm a lay person, by the way. And also, once these therapies are proven to be helpful, will insurance companies pay for them?
Yeah.
How's that gonna happen?
Let's talk about the first question first, because the second one is more political. So, N of 1 is really challenging. I've been struggling. It's this is where you end up having more opinion than people, because there is lots of efforts on precision-based medicine, N of 1 therapeutics. There's different consortia now. There is an N of 1 collaborative in the United States. There is the, what is it called, 1M in the EU, which is similar sort of a academic collaborative for people to really try to tackle this issue of N of 1 approaches. What is necessary to go into the patient? And I have a personal opinion here, because I can't say that represents the field, because again, there's really more opinions than people here. And I have a hard time with the N of 1 approach because of all the issues I mentioned about placebo, not really knowing. But I mean, there are certain diseases where we know what's going to happen. If I have a boy with Krabbe disease and it's 2-month-old baby and is already having symptoms, I know what's going to happen to that kid. I know that the course of the disease, right? So in that situation, I also know what I could be looking for to see if what I'm doing is actually helping, right? But if I don't know what's going to happen to that patient, if I don't know what I'm going to be looking for, and just blindly say, let's do this because we don't know what else to do, I don't know if we're going to, for one, we will never find out if we actually help that person, right? And we may have increased harm into that person. So I'm challenged with that approach. I want to know what I'm looking for. If I want to have what we call an outcome measure. So it's really, whether you do a trial in 10 people, 100 people, or in one, you still need to have an outcome measure, and you need to have a baseline assessment, and you have to have an anticipation as to how that changes. I think that has to be in place.
Variants, you know the precise variant, and you can treat it so personalized to that individual. That's what I'm wondering about.
Yeah, and we are talking exactly about that. We're talking about a personalized approach where you're going and say you're going to correct the gene defect, right? The problem is that diseases are complex. And so, for example, we know, for example, again, taking ALD as an example, if you do gene therapy for ALD, if you do it past a certain time point, it's not going to work. And that's, we say the horse is out of a barn kind of situation. When you start having a downstream effects of that mutation that take on their own life, and become their own sort of pathophysiology, you may go on top and fix that, but it's too late, and you're not going to change anything. And so that's why you need to know what you're trying to change. You need to have something that you can measure in the patient to see if it's actually helping. And then the other approach, the other piece with the N of 1, the challenge that we have, which can be overcome, is to figure out whether that theoretically designed product that should increase your gene expression actually does it in the cells that you're going to be doing it for. Because, people say, oh, it's easy. We'll just take your skin cells and we'll develop a drug in the skin cells, and then we're going to inject it back to you. Well, it may work in the skin cells, but that doesn't mean that thing is going to ever get to your brain, right? And so with all the challenges we just discussed. So these are the issues that really need to be sorted out. But I think what is going to happen, I'm hoping in the next 10 years, is platform-based approaches to this, where you develop a transporter, a vector, right, that we know is working well to get it into these type of cells. And then basically all you need to do is just swap out the gene. And so that it could become personalized medicine from the perspective of each person may have a personalized product, but the approach, how you give it to them, the anticipated side effects, and the way you're going to measure how they're responding is the same, right? And so that will be a so-called platform trial. So I'm really hoping that we're going to get there. And technically, actually, we are there. But the regulatory agencies are not yet on board. They need to wrap their head around it. And they are realizing that. So FDA, for example, has been now putting a lot of effort on trying to think through what are we going to expect from a company or from a sponsor to do a platform type of trial. So I think it's coming. I really think it's coming. And I'm more excited about that approach, to be honest, than we're going to make this one ASO for you, and this is going to work, because I see too many challenges there. But that's my opinion, and I think others don't share that opinion. Just so you know. Yeah, so the primary right now, when people talk about NF1 therapy, most of the time they're talking about the personalized ASO, antisense oligonucleotide. But I think with gene editing becoming an availability in the next 10 years, that means you're going to go and correct the DNA defect. And again, every disease has many different mutations usually. Then it will become more, that's where the platform, so the strategy will be needed. But right now, I think most of the companies that are there that are pitching the NF1 approach are ASOs. I don't know of any gene editing company yet that has been, maybe there is, I don't know. I haven't heard of them yet. There probably is.
Maybe a stupid question, but you describe these different techniques, and I mean sometimes we have the same disease and we have approaches that go over ASO or via gene therapy. How would you foresee what will be the future? So will we in future just have one of the techniques which is kind of outstanding and the best one, and we do everything with it? Or can we stratify that this disease is better with this technique, or what do you think will happen?
Yeah. So your question is directly connected to your second question that I didn't answer. And that's who's going to pay for it. And I think that's going to be critical. The cost of these therapies are going to be a major determinant of what is going to be realistic, right? Because we are currently living in a world where, if you can afford it, you're going to get cured. If you can't afford it, you're going to die. And maybe there are now the interims. This therapy is not as good as that one, but it's more affordable, right? And so this is terrible. But again, like I said, this is more politics and policy than reality. I mean, the reality is that for each disease, it's going to be different. And in certain diseases, actually gene therapy is going to be better. In others, it may be actually better to use an ASO, because we are learning. As we are learning these new things, we might see that perhaps you need an intervention for a finite amount of time rather than a lifelong treatment. So remember, the gene therapy, you sort of have this irreversibility that you cannot go back, right? You cannot remove that gene from the body once it's in there. And so I really think it depends on the disease. It depends on the stage of the disease as well, right? So like you may have certain stages of the disease that may respond better to ASOs, and other stages that may respond better in a gene therapy, right? And the ideal future would be that all of this becomes really preventive, right? That we do this before the patients have anything, And so if we think from a prevention strategy, ultimately we think gene delivery is probably the best, right? Or gene editing is the ultimate best approach. So I think this question is going to change based on that. But I think also that those are going to be the most expensive, like a personalized medicine of an NF1 that means it's going to cost millions per patient, right? And so these things are expensive, like doing these experiments in the lab to just figuring out if something is working and then doing toxicology on them. These are very expensive studies. And so cost is really an unaddressed issue. And it's definitely above my pay how to solve the cost problem. I think that's a societal problem that's emerging. Yeah.
Type of trials are more prevalent or done more. Do you think that will help
Yeah.
that?
That's the whole, I think that's part of the hope, is that once these sort of platform approaches are accepted, the cost of the production is going to be significantly falling down, and the cost of the development is going to go down. Because right now, if I give you a gene therapy with this DNA sequence, I have to go and do toxicity studies in 2 different animal species, and that can be millions. And the question is, could we say, well, rather than having to do every single time you change a single base pair, a whole set of 2 different species of animal studies for 2 years, could we just do, one big study, and then for each variation, maybe do a much shorter, smaller toxicology study in one single species, maybe in mouse brains. So that would take only perhaps a few months rather than like several years, to get to a result. So that would decrease the development costs. And then the big factor is the production. Right now, actually, we are struggling with the approved products that are already on the market to get enough production of the viruses. And so viral production is expensive and it's very difficult, and it's done only in very few facilities in the world right now. And so that's why having these sort of platform approaches where you have this as a factory in the future could really make a difference to the cost. So that's the hope.
Yes. Could it also help you bring in more effort to find help for those people that are further into their illness, where normally before, I've heard you say a couple times, once you're past a certain age of illness, it's harder to treat.
Yeah. I don't think a platform trials are necessarily make a difference there, right? Because they won't affect pathophysiology in a different way. But I think that because of right now we have about, I want to say around 8,000 different genetic diseases. Okay, we have 24,000 genes. We know at least 8,000 of them can go wrong. There probably can be all of them going wrong. So there are probably 24,000 different genetic diseases. And we know about 5,000 of them affect the central nervous system. So it's actually a huge number of genetic diseases affecting the brain. And so one of the hopes with the platform trial is getting access for patients that have an ultra-rare disease. That's more the hope, rather than like within the disease group to try to go into later stages of the disease. That's just always going to come down to the mechanism of the disease, so the pathophysiology of that disease. Like I said, there are some diseases where we know when a cascade of events is triggered, we can't stop it. And it's literally like the horse is out of a barn. And so, yeah.
Talking about is an ultra rare disease.
Yeah.
Very few under 100 for sure, probably under 20 that I know of. But so there's not a lot known yet.
Yeah.
So that's what I was wondering, if these platform trials will help us for these types.
That's the hope. Yeah.
Okay.
So the thing is that if we still need to know about the natural history of the disease, we still need to know what we're going to be looking at, right? To what do we want to get better, right?
Yeah.
But at least from a production machinery perspective of the product, that those costs are going to go down. So
Okay.
that will make it more accessible. Yeah.
Thank you.
Just real quickly, the FDA actually has an open comment period right now, whether or not to accept what Dr. Padini is talking about, platform technology. So if you go to the FDA's website, and want to offer a public comment, you can do that.
Awesome.
Happy to give you that link.
I would
Right.
love it.
Thank you. Okay, I think that's it. Any more questions? I'm around for the rest of the day, so thank you.